Hello and welcome back. We're going to continue talking a little bit about the flow of information, genetic information in the cell. So the next step is going to be translation. Translation is going to take an RNA molecule and it's going to take the information in those nucleotides and convert it into protein, okay? So in this case, we're going to go from a nucleotide to an amino acid. And that process of taking the information from RNA and converting it into protein, that's translation, okay? So it's translating that message, and the way that the cell is going to do that is going to be at the ribosome, okay? So translation is going to happen out in the cytoplasm. It's going to happen out at the ribosome. And what we're going to have is we're going to have our messenger RNA with our cap, and I'll make that as a box. And then we're going to have some sequence that makes up our messenger RNA. And I'm going to put an example sequence up here on the board. All right. Then we've got our poly-A tail, all right, on our messenger RNA, because those were things we talked about in the last video with splicing. Now, the way the ribosome is going to read that messenger RNA, the messenger RNA is going to enter the ribosome. That cap is going to be at our 5' end. The poly-A tail is at our 3' end. That ribosome is going to scan along until it reaches the first codon. So the way this is going to be organized and the way the ribosome is going to read these nucleotides are going to be in 3-unit blocks, okay? So we've got 3 nucleotides together. Those 3 nucleotides are going to equal a codon. So 3 nucleotides is going to give us a codon. And the reason that the genetic information has to be read this way is because we have 4, oops, that's not an E, we'll make that a U. We've got 4 nucleotides that make up RNA, right? We've got adenine, uracil, cytosine, and guanine, all right? But we have 20 amino acids, okay? 4 definitely does not equal 20, okay? So we have to put the nucleotides in some combination in order to generate enough combinations that we can insert each of the 20 different nucleotides, okay? So because we have 3 nucleotides to make 1 codon, what that gives us are 64 possibilities. So we've got 64 possibilities that can come out of putting 3 nucleotides together. And if you want to go through the mental exercise of trying to come up with all of the combinations of A, U, C, and G in 3-letter combinations, you're welcome to do that. Or you can look in your PowerPoints because there's a table there that already has that information figured out. So that table in your PowerPoints that I've got listed as the genetic code, I've got the Rosetta Stone as a reference there for it, that is going to be those 64 possibilities, okay? And what we're going to have with those 64 possibilities is the ability to put in any of the 20 amino acids, okay? Now you'll notice with that genetic code there's going to be what's known as some redundancy in there. And that just means, what that redundancy means is sometimes you have more than one codon specifying a particular amino acid. Okay? Now in some cases on there you're going to have just one codon that specifies just one particular amino acid. And that's okay too. But the redundancy we've got there, you've got more than one codon that specifies a particular amino acid. Okay? We've got some special codons within the genetic code, and those special codons are going to be the ones that are going to start and stop the process of translation. Okay? So one of our special codons is the AUG. AUG will insert an amino acid. It will insert a methionine. Okay? But this is also where the ribosome knows it needs to start building a protein. Okay? So if you notice here with my first codon that I put in, I have the sequence AUG. Okay? Because it's going to read that information in the cap. The cap won't have an AUG sequence. When it reaches that first AUG codon, that signals the start of translation. And that'll insert our first amino acid. That'll insert our methionine. Okay? Now, again, I don't want you to memorize that big code table in your PowerPoints, but what I do want you to be able to do is use it. So in quizzes, in tests, I will provide you with that table, but you have to know how to be able to use it. And we'll go over that here in just a little bit. Okay? But before I get to that, I'm going to talk about the other three special codons. The other three special ones are going to be UAA, UGA, okay, and UAG, okay? Those three codons are going to specify stop, okay? And in this case, you're not going to insert an amino acid. It's just going to leave an empty space, and that's how it's going to signal we've reached the end, okay? So with the short RNA sequence that I put up here, if you notice, I've arranged it so we've got nice codons here. We're kind of going to go through how to identify those different codons. The example that I've got in the PowerPoint, all right, is using the codon UAC, all right? That UAC is going to encode specifically for the amino acid tyrosine. And the way we're going to do this is we're going to follow our table using each of the base pairs. Okay? So we've got first base, second base, third base here. All right? Unfortunately, it's not baseball. We're talking about nucleic acid bases here. All right? On the left side of that table, you're going to find the U on that table. Okay? That's going to narrow things down to a column. Then you're going to go to the top of the table and look for the A. That's going to narrow your column down even further because it has to overlap between the U and the A. Then you're going to go to the right-hand side and look for the C. Whatever is next to that codon, UAC, is the amino acid that will be inserted. In this case, it would be tyrosine. If we do this again with our sequence, all right, if you track CCC, that's going to insert a proline as the amino acid, all right? If you go AAA, that's going to insert a lysine, all right? And your UGA, this is one of our special ones. This is one of the stops, okay? So that would not insert any amino acids. So in this case, we've got a very short protein. We've got a 3 amino acid protein that consists of methionine, proline, lysine. Now I'm giving you the full names of it. I think the table just has the abbreviation, the 3-letter abbreviation. I am absolutely fine if you just fill in the 3-letter abbreviation. Don't worry about that, okay? What I want to be able to see you to be able to do is what I just did here on the board. And that's take those codons, find the corresponding amino acid, build the protein, okay? Now, with our genetic code, we've got the redundancy, we've got some special codons in there that are defined here, our start and our stop. The other thing with the genetic code, we're going to have a little bit of what's known as wobble in the genetic code, okay? And that wobble is at our 3rd base. The ribosome and the RNA, when that comes together, the first 2 bases sit there in the ribosome very, very securely, all right? The 3rd one, it's a little bit wiggly, okay? And that's why, in part, that we've got the redundancy in the genetic code that we do, because if you look at proline as an amino acid, you will notice that there are 4 codons that will specify the amino acid proline. CCC, CCA, CCG, CCU will all specify proline, okay? And that essentially accounts and allows for that wobble so that you don't get a different amino acid inserted, okay? So with our lysine, all right, our lysine's a little bit different. The AA is consistent with our lysine. The other codon that encodes lysine is AAG, okay? But if this becomes an AAU, you're now going to insert a different amino acid. This is how some mutations can happen in proteins, how you get some changes, because that 3rd base is a little bit wobbly, okay? It just doesn't set in the ribosome. At least this is the way I think about it. It doesn't set in the ribosome as securely. But because we have the redundancy in the genetic code, a lot of times the correct amino acid is inserted regardless of what the 3rd base is, okay? As long as the first 2 are correct, all right? So keep that in mind, all right? We've got redundancy, we've got our special nucleotides, we've got a little bit of wobble that can happen. The next video, I'm going to describe a little bit about the physical process of assembling the protein at the ribosome. There are also some videos that actually are animated that are probably going to show that better than what I will draw it, but that's okay, all right? We'll go over that anyway. All right, now in terms of tests and quizzes, like I said, what I want you to be able to do is take the information from the RNA, tell me what protein would be made, okay? Or what amino acids would be inserted to make the protein. All right, I've got a homework assignment to let you practice that. If you have any questions, please let me know.